Welcome to the Deep Dive, your fast track to expert-level knowledge. Okay, so today we're really getting fundamental. We're diving into the raw material of evolution itself, genetic variation.
That's right. It's the absolute starting point.
We're going to unpack how the differences coded in DNA actually, show up as the traits we see in organisms.
And it's crucial. The famous biologist Ernst Mayr put it bluntly, without variation, evolution is just, well, it's meaningless. It can't happen.
So our goal today is to look under the hood, right? Go beyond just saying mutation and explore the actual ways life gets new genetic information.
Exactly, the whole toolbox. And based on the source material, it's a pretty packed toolbox.
Yeah, it seems like it. Even setting aside something huge like endosymbiosis, creating eukaryotes, the chapter points to 5 main ways. Things like multiple alleles, changes in chromosome numbers.
Changes in how genes are regulated, those jumping genes or transposons, and even genes moving between species, horizontal gene transfer.
That variety of mechanisms really feeds into some big, still unresolved questions in evolution, doesn't it? The source highlights 4 key ones.
It does. First up is that huge puzzle, the genotype-phenotype connection. How does the DNA blueprint translate into the actual organism? Sometimes nearly identical genes give really different results. That's the heart of Evo-Devo, evolutionary developmental biology.
Okay, what's the second big question?
It's about weighting. What's driving more evolutionary change? Is it the small point mutations, or is it these subtle shifts in gene regulation turning genes up or down?
Right, and the third?
Looking beyond mutation and regulation, what about other sources, like gene flow, mixing between populations, or just random chance genetic drift? How important are they?
And finally, the environment's role.
Yes. The fourth question is how ecology and development, the organism's context, actually shape how much variation gets expressed in things like body size or lifespan.
Makes sense. So where should we start this exploration? Maybe at the big picture level, chromosomes.
Let's do that. Start big. Variation isn't always just changing one letter in the code. Sometimes you copy the whole instruction manual.
You mean duplicating entire sets of chromosomes?
Exactly. That's polyploidy, having multiple complete sets. And it's incredibly common, especially in plants. Some estimates say 40, even up to 70% of plant species are polyploid.
Wow. Why? What's the advantage of having all those extra chromosomes?
Well, it can do a few things. It can increase the size of cells, sometimes the whole organism. That might be good on its own, but maybe more critically, it adds stability and just a massive potential for genetic shuffling, for recombination.
Ah, so if the environment changes.
You've got a much bigger genetic toolkit to draw from, more combinations possible, better odds of hitting on something useful.
And the classic example driven by us humans, really, is wheat, right?
Yeah.
The source walks through its history in the Fertile Crescent.
It's a perfect case study. The story of bred wheat, Triticum aestivum, it starts way back with a cross between 2 wild grasses, each with 14 chromosomes, einkorn wheat, and a type of goat grass. They hybridized naturally, creating a 28-chromosome hybrid, which we call wild emmer wheat.
Which people then started farming.
Right, and then maybe 9,000 years ago or so, this cultivated emmer wheat crossed again, this time with another 14-chromosome goat grass species.
So adding another set of chromosomes.
Precisely. That gave rise to spelt, which is an allohexaploid. That means it has 6 sets of chromosomes from 3 different ancestors, totaling 42 chromosomes.
Hexaploids, 6 sets. Okay, so that's a combination of different genomes. And then how did we get to modern bred wheat?
That last step was actually genetically relatively minor. It was a single mutation that made the seeds easier to thresh, to separate from the husk without breaking the whole seed head.
Ah, a practical farming advantage.
Made a huge difference for agriculture, allowed bred wheat to really spread.
And this isn't just something ancient. We've seen it happen in the lab too, haven't we? With tobacco.
Yes, the Nicotiana degluta experiment. It's proof of concept, really. Researchers crossed 2 tobacco species, N. tabacum with 48 chromosomes and N. glutinosa with 24.
Okay, what happened?
They got a hybrid, but it had 36 chromosomes, an uneven number, and it was sterile. The chromosomes couldn't pair up properly for reproduction.
Made sense. But then,
just spontaneously, in one of these hybrid plants, the chromosome number doubled, went from 36 to 72.
And suddenly everything had a partner.
Exactly. It became instantly fertile. It was reproductively isolated from the parents. Boom, a new species, N. degluta, created right there through polyploidy. Speciation in a single step.
That's incredible. And this theme of ancient duplication isn't limited to grasses. The source mentions apples, too.
Right, the common apple, Malus domestica. Genetic evidence suggests its ancestor underwent a whole genome duplication event way back maybe 50 million years ago.
And that changed its chromosome number.
Yep. Went from an ancestor with probably 9 chromosomes to the 17 we see in apples today.
And did that duplication do anything specific for the apple?
It seems so. It's linked to the evolution of a particular family of genes called MADS box genes, and changes in those genes are thought to be key in developing the pome fruit structure, the fleshy part we eat.
Madbox genes. What do they generally do? For those of us not deep in plant genetics.
Fair enough. Think of them as master regulators, transcription factors. They control how structures like flowers and fruits develop. So duplicating the whole genome gave this AdMadbox family extra copies.
Which could then evolve new roles.
Exactly, neofunctionalization, leading, in this case, potentially to a whole new kind of fruit structure.
Okay, so duplicating the whole genome is one major source of variation. What about rearranging the parts you already have? Changes within chromosomes.
Right, now we're talking about changes to the structure, the phenotype of the chromosome itself, things like deletions, where bits are lost.
Usually bad news, I imagine.
Often harmful, yeah, especially if you only have 2 copies, like in diploids. Maybe less so in polyploids with backups. Then you have duplications, bits of extra material.
Where do they come from?
Often from mistakes during meiosis, like unequal crossing over. One chromosome gets an extra copy, the other loses it. And these duplications are really important. They create redundancy.
Like those MADS box genes again. An extra copy that's free to change.
Precisely. It's a major engine for evolutionary novelty, creating gene families. Then you have inversions.
Where a segment flips around, I've heard them called supergenes.
That's a good way to think about it. An inversion can lock a set of genes together. Because the inverted segment can't easily recombine with the original version, beneficial combinations of alleles within that block can be inherited as a single unit.
Protecting a good combo, basically.
Sort of, yeah. And then there are translocations.
Moving pieces between different non-homologous chromosomes. The muntjac deer example here is wild.
It really is astounding. You compare the Indian muntjac and the Chinese muntjac. They look pretty similar, same amount of DNA roughly, but they're chromosomes. Completely different.
How different?
The Indian muntjac has just 3 pairs of huge chromosomes. The Chinese muntjac has 23 pairs of smaller ones.
Wait, 3 pairs versus 23. How did that happen?
It seems the Indian lineage underwent a series of translocations, probably Robertsonian fusions, where smaller chromosomes essentially got stuck together end to end, creating the larger ones.
That's massive structural change, happening relatively quickly in evolutionary terms.
It shows how dramatically genomes can be reorganized, not just point mutations, but huge rearrangements.
And we can track these kinds of changes using techniques like chromosome banding, right?
Yeah.
Especially in primates.
Absolutely. G-banding stains chromosomes in a specific pattern of light and dark bands. When you compare human chromosomes, we have 23 pairs, to chimps or gorillas, they have 24 pairs.
The patterns are mostly identical.
Almost entirely. The banding patterns line up beautifully for most chromosomes, but there's one big difference. Human chromosome 2.
What about it?
It clearly corresponds to 2 separate chromosomes found in chimpanzees and other apes. The banding pattern shows it's an end-to-end fusion of those 2 ancestral chromosomes.
Wow. So that fusion event explains why we have 1 fewer pair than chimps.
Exactly. It's incredibly strong physical evidence for our shared ancestry and that close relationship with chimpanzees, a direct historical marker in our DNA.
Okay, that locks it down pretty well. So, moving from whole chromosome structures down to the level of individual genes. Let's talk about gene duplication again, but focusing on divergence.
Right. And here we need to be clear on some terms the source uses, paralogs and orthologs.
Yeah, help us keep those straight.
Okay, think of it like family history. Paralogs are like related genes within the same species, arising from a duplication event in that lineage. Good example. The different globin genes in humans, alpha, beta, gamma hemoglobins, they all came from ancient duplications but now have slightly different jobs.
Okay, paralogs are internal duplicates. What are orthologs?
Orthologs are the same gene in different species, inherited from a common ancestor. So the alpha hemoglobin gene in a human and the alpha hemoglobin gene in, say, a horse are orthologs. They trace back to the same ancestral gene before humans and horses split.
Got it. Paralogs within, orthologs between. And the globin family itself tells a story of these duplications over time.
A really clear one. By comparing the amino acid sequences, how similar or different they are, we can build an evolutionary tree for the globin genes.
What does that tree look like?
Well, myoglobin, which stores oxygen in muscles, seems to be the oldest branch. It diverged first, then came the split that created the ancestors of the alpha and beta globin chains.
Which form the main part of hemoglobin.
Right, and the most recent split shown in the source material is between the beta-like chains, the delta and gamma hemoglobin genes. They are still very similar, indicating a more recent duplication.
So when a gene gets duplicated like this, what can happen to that extra copy? It doesn't always stick around or become useful, right?
No, there are a few main fates. One is that it's just redundant, doesn't offer an advantage, and it starts accumulating mutations. Over time, it can decay into what we call a pseudogene, basically genetic fossil, non-functional.
Okay, so it can become junk. What else?
The more interesting fate is neofunctionalization, neo meaning new. The duplicate copy evolves a completely new function, different from the original gene.
Any examples?
A classic one is crystalline proteins in the lenses of our eyes. They help focus light. But evolutionarily, they actually derive from mundane metabolic enzymes that got duplicated and repurposed for this entirely new role in the eye.
From enzyme to lens protein, that's quite a career change.
Isn't it? And there's a third possibility mentioned, a bit more complex, seen in honeybees, Apis.
What happened there? Did the duplicate take over?
Sort of. It's about the fem gene, which controls sex determination. It got duplicated, creating a sister gene right next to it called CSD. Now, CSD took over the main job of determining female versus male development.
Okay, so what happened to the original fem gene?
Because CSD was handling the critical function, the selective pressure on fem relaxed. It wasn't quite as essential anymore. This actually allowed fem to accumulate more nucleotide variation than it otherwise would have.
Ah, so the duplication, by providing a backup, indirectly increased variation in the original gene.
Exactly. The redundancy effectively bought the species more evolutionary options at that spot. It's a subtle but important effect.
That's fascinating. Okay, so we've covered big structural changes, gene duplications. What about the really molecular level drivers? The transposons and gene regulation?
Right down to the nuts and bolts now. We absolutely have to talk about transposons, those jumping genes, sequences of DNA that can move themselves around the genome.
And this is where Barbara McClintock comes in.
Absolutely.
Yeah.
Her work in maize back in the 40s and 50s, identifying the H&Ds elements that were moving and causing changes in kernel color, it was revolutionary.
But not immediately accepted, famously.
Not at all. The idea of a dynamic, mobile genome went against everything people thought they knew. She actually stopped publishing on it for years before finally getting the Nobel Prize in 1983, just way ahead of her time.
So how do these things actually move?
The mechanism varies, but a common way, like for the IS1 element in E. coli, which is similar to how some work in maize, involves the transposon making staggered cuts in the target DNA, inserting itself, and then repair enzymes fill in the gaps.
Well, they can spread really fast.
Incredibly fast, especially if they hitch a ride via horizontal gene transfer moving between species. The example given is the P transposon.
What happened with that?
It seems it jumped from one species of fruit fly, Drosophila willistoni, into the main lab species, D. melanogaster, maybe only 50 or 60 years ago, and now it's found in basically all wild populations of melanogaster worldwide.
That's a rapid invasion across a whole species.
Yeah.
And these elements are common in us too.
Oh yeah, the ALU sequence, for example. It's estimated there are over 1 million copies scattered throughout the human genome. They make up a significant chunk of our DNA.
Wow. So they're moving around, inserting themselves. That's a source of variation right there. But then there's the whole layer of controlling the genes we have. Regulation.
Crucial. Having the genes is one thing. Controlling when and where they're turned on or off is maybe even more important for evolution. This is the regulatory layer.
And the source mentions cis and trans regulation. What's the difference?
Think location. Cis regulation involves control elements, DNA sequences that are physically located near the gene they regulate, often right next to the promoter region. Changes here are thought to be really important for evolving different body shapes, morphology.
Okay, local control. And trans.
Transregulation is distant control. It involves factors, usually proteins called transcription factors, that are encoded by genes elsewhere in the genome. These factors travel through the cell and bind to specific sites, like the TATA box near a gene, to turn it on or off.
Right. And regulation doesn't stop when you start making RNA, does it? There's post-transcriptional control too.
Definitely. A single gene's initial RNA transcript can be processed in different ways. Alternate splicing is a big one.
Where you snip out different bits to make different final messenger RNAs.
Exactly. One gene pre-mRNA can lead to multiple different proteins. The waxy gene in rice is given as an example. Then there's RNA editing, changing bases in the RNA itself.
And then there's this whole world of small RNAs involved in regulation, RNA interference.
Yes, RNAi. Huge discovery, won the Nobel Prize in 2006. It involves small interfering RNAs. They essentially act as guides to target specific messenger RNAs and block them from being translated into protein, often by chopping them up.
Like a targeted silencing mechanism.
Very much a cellular defense and regulation system, but maybe even more widespread in their regulatory role are the microRNAs or miRNAs.
miRNAs. Tiny, right?
Really small, just 20 to 22 nucleotides long. They don't code for proteins themselves, but they bind to target messenger RNAs and trigger their degradation or block their translation.
And they regulate a lot of genes.
An enormous number. The estimate mentioned is that they could be involved in regulating a third or even more of all human genes. They are major players in the regulatory network, sometimes called killer RNAs for their effect on mRNA.
Wow. So there's this whole complex web of control happening after the gene is transcribed. And the source also mentions piRNAs, protecting the genome.
Right, Piwi-interacting RNAs or piRNAs. They seem to be particularly important in germ cells, sperm and egg precursors, acting as a kind of defense system to silence transposons and prevent them from jumping around and messing up the crucial genetic information passed to the next generation.
Okay, so pulling all these threads together from whole genome duplications in wheat to chromosome fusions in our own lineage, gene families diverging, transposons jumping, and these tiny regulatory RNAs like miRNAs. What's the big picture message for the listener?
I think the core takeaway is that genetic variation isn't just about slow, random, single-letter typos. It's a really dynamic, multi-layered process. Evolution uses everything from massive structural rearrangements down to incredibly subtle regulatory switches.
It's a much more active and complex system than just random mutation.
Absolutely, and understanding all these mechanisms, the big polyploidy events, the translocations, but also the alternate splicing, the miRNAs, the transposons, is key to seeing how populations can actually generate novelty and respond, sometimes quite quickly, to selective pressures.
And for final thought, the source brings up something really intriguing connecting stress and variation, involving those piRNAs and a heat shock protein.
Yes, this is a really provocative idea. We know piRNAs usually silence transposons in germ cells, but there's evidence linking the heat shock protein HSP90, which helps proteins fold correctly, especially under stress.
Okay.
Linking HSP90 activity with piRNAs in a way that, under stressful conditions like heat shock, might actually release the silencing effect on some transposons.
Wait, so stress could actually activate jumping genes?
That's the implication, that environmental stress might not just be a filter-selecting variation, but could, through mechanisms like this involving HSP90 and piRNAs, actively unleash new genetic variation by allowing transposons to move.
So the stress itself could be triggering the generation of the raw material needed to potentially adapt to that stress.
It's a fascinating feedback loop idea, that the potential for variation might be hidden or buffered, and stress could unlock it. A really exciting area for future research, connecting environmental pressure directly to the generation of novelty.
That definitely gives us something to think about. Thank you for walking us through that complex landscape of genetic variation today.
It was my pleasure. Lots of moving parts.
Indeed. For more deep dives that get to the source of the science, keep listening to The Deep Dive.